Just thought I might add that.
All right, I believe we have a critical mass, so let's get going. We are in for a treat today. Muyiwa Awoniyi is one of our own who has done exceptionally well during his combined research and clinical GI hepatology fellowship. Mu has a diverse background. He majored in science with a minor in music. So he threatened to hum the good, bad, or sing the good, bad, and ugly for us, but deferred. So he then spent several years at Mayo Clinic in the Department of Biochemistry and cell biology. So he has a very strong background in basic and molecular biology. He then got a combined MD PhD degree at University of Washington, Seattle. He's taught me that this is UW to the insiders. And then completed internal medicine residency at University of Michigan prior to being recruited to our T32 program and basic science pathway for GI hepatology fellowship. He enjoyed the lab experience so much, he stayed for an extra third year under support of the immunology T32 and the microbiology immunology department. So he's extremely well grounded in basic science. Then completed a clinical GI fellowship and currently is about to complete a UNC liver transplant specialty fellowship. He's board certified in gastroenterology, internal medicine, and board eligible in liver specialty. Mu has embraced his research with one of the most impressive amounts of enthusiasm and endurance that I've experienced in research fellows, and has been very effective in starting a program from scratch in which he did not build on an existing program in my lab, but really created his own new models that he will tell you about today, and generated a whole new body of novel research that I think will is the first component of a sustained, very productive, independent research career. He's in the late stages of job search with quite a bit of a bit of enthusiasm among some stellar programs around the country. So, Mu, I want to turn it over to you to tell us about the good, the bad, and the ugly resident microbes that drive PSC-related liver disease.
Thank you, Balfour, for the kind invitation, and thanks for everybody being here. I have no relevant financial disclosures. I plan on breaking up my talk into two parts. One focused on PSC and my PSC model, and the last sort of small bits about the PSC-IBD model. Regarding the first part, the majority of the talk, I'll give a background on what primary sclerosing cholangitis, or PSC, is, and talk about the burgeoning data regarding the microbial association with PSC, and then go into greater detail about my murine models, where I explore potential microbial-driven mechanisms, and then sum up with future directions as well as clinical impact, and then take the last part talking about the PSC-IBD model and its relationship not only the colitis, but also colorectal dysplasia. And a lot of people will tell you, when particularly physicians, when they think about a problem, I think there is a face to it. And at least the face to PSC is at least was for me sweetness, and is for me sweetness. And for those that don't know who Sweetness is, he is Walter Payton, probably the best running back. He plays for the Chicago Bears, my hometown, and I grew up watching him. The impact that he made on the field was even much more off the field and what he's done for urban city Chicago. And I didn't know at that time that this young man that had all the resources of the world, but died from a complication of primary sclerosing cholangitis in the form of cholangiocarcinoma or bile duct cancer. That, you know, this is the presentation where we have a lot of young people that we have nothing to offer, similar to him, despite his wealth and his resources. And so with that, talk about primary sclerosing cholangitis. It's, or PSC, it's an idiopathic chronic and cholestatic inflammatory liver disease with a variable course and typically insidious presentation. In some cohorts, it can recur or progress to develop hepatobiliary fibrosis. And in up to 10% of people, it develops, you can develop cholangiocarcinoma or bile duct cancer. Gold standard for diagnosis is usually by cholangiopathy, and biopsy is used to diagnose small duct disease or overlap syndromes. It has 1 to 16 per 100,000, and it's typically male predominant with a mean age of 30 to 40 years old. Again, this is a very young person's disease. Along with that, it has a very strong association with inflammatory bowel disease, with nearly 5 to 7-fold increase of risk, and showing the standardized mortality rate over time per age group is important. Not only that you have an increased predisposition to develop IBD, but death is associated largely to young people. And as you can see here, either 1, 5, or 10 years, up to 6-fold increase in individuals with PSC-IBD less than 40 years old, just based on that age. So it is a disease of the young that we have very little regarding medical therapies to offer. And even for those that progress to need a liver transplantation as a potential option, disease reoccurrence is up to 30%. So it doesn't end there. So some people believe that PSC is hepatology's greatest black box, in which I come to agree. Part of the reason for the lack of therapeutics is based on the unclear nature of the pathogenesis of PSC and its link to IBD. The impact of genetic susceptibility factors on overall PSC liability is less than 10%, while environmental risk is likely accounts for more than 50%. Furthermore, reoccurrence of PSC after liver transplantation, as I said, supports the idea that factors outside the liver itself may drive disease development. A plausible theory of PSC and colitis involves the altered microflora or dysbiosis, producing potential toxic immune byproducts that translocate to the liver and induce hepatobiliary inflammation and injury, presumably through a pathway that involves the activation of innate and adaptive immune responses in genetically susceptible hosts. Moreover, there's increasing evidence to support a link between PSC and resident microbial populations. Overrepresentation of intestinal pathobionts have been seen in PSC patients in multiple studies, including Enterobacteriaceae such as E. coli or Klebsiella pneumoniae, Enterococcus efficalis, or the family Veillonelliaceae, Veillonella, that's been found to overrepresent and have some potential associated with pathobiont type behavior. And then additionally, a recent study in gut by Schramm's group showed a predominance of biliary efficalis compared to controls, suggesting potential translocation. In 2020, a publication out of Gastro showed that in a fecal metagenomic study, the largest performed by Hav and Kuman, showed increased fecal E. faecalis, but also reported a decreased abundance of protective bacteria, Lactobacillus, which I'll talk about more. And then there's multiple studies that have reported that colectomy before liver transplantation had been associated with reduced recurrence in PSC. So how I conceptualize microbial influence in PSC is in maintaining a homeostasis by striking a balance between injurious as well as protective resident microbiota. In the following slides, I will provide you additional experimental and clinical studies suggesting bacterial influences in both arms. Let's talk about back in the 90s. One of the first papers that I read regarding this field was out of Balfour's lab, in which there were rat studies that showed translocation of bacterial byproducts, notably peptidoglycan and LPS, from a leaky luminal mucosa in a self-fulfilling blind loop rat model that leaked into the portal circulation, leading to hepatocyte necrosis and small duct biliary inflammation, very resembling early stage of PSC, that was attenuated with treatment with metronidazole and gentamicin, resolving disease. Again, suggesting a pathogenic role for the microbiota. Over the last decades, there's been several studies that have shown possible protective roles of antibiotics in PSC. Here I present a meta-analysis by Shaw, in which looks at a total of 124 PSC patients over multiple studies, receiving antibiotics, which include vancomycin, minocycline, metronidazole, as well as rifaximin. Treatment with antibiotics in PSC was associated with statistically significant reductions in short-term biochemical outcomes. Vancomycin appeared to be the most effective of the antibiotics, but it had several limitations. There was a paucity of placebo-controlled trials. Out of the five studies that I presented you, 3 of them were randomized in very, very small trials. There were no pre or post-therapeutic microbial analysis, so we don't know what the antibiotics are targeting. There were small studies with moderate heterogeneity, suggesting an overestimation effect. Safety profile, standard dosing, and long-term studies were lacking. And then there were a lack of hard clinical outcomes, such as mortality or survival-free liver transplantation. And although alkaline phosphatase is a strong surrogate biomarker for PSC, it has not been linked with hard outcomes. In some, there's promising efficacy signals from long-term antibiotics in treatment in PSC. However, further studies are needed to clarify the mechanisms responsible for the observation, which led AASLD to put out a guidance this past fall that there are currently insufficient evidence to recommend the use of oral vancomycin outside of clinical trials. So there are also studies to support a protective role for the gut microbiota in PSC, which includes small studies such as this one by Dr. Barodi in 2013, and where donor FMT in a PSC-IBD patient reversed both hepatobiliary as well as intestinal disease, shown here, where before and after, where it moved from a Mayo 3 to a Mayo 0 disease, suggesting again a protective role for the gut microbiota. In 2016, a study out of Mayo showing worsening liver disease in a germ-free mouse model of PSC, the MDR2, which would suggest again an overall protective role for the gut microbiota, as shown here with conventionally raised versus germ-free, looking at alkaline phosphatase, ALT, total bilirubin, serum bile acids, all of which were worse in the setting of germ-free, in a germ-free state. So this leads me to hypothesize that there's a functional balance of resident intestinal bacteria facilitating hepatobiliary inflammation and fibrosis by production of protective and detrimental byproducts. with my aim of identification and validation of protective and detrimental bacterial species and metabolites mediating hepatobiliary inflammation and fibrosis using the MDR2 knockout mice, which I'll go over in the next slide. So the MDR2, or the multi-drug resistant gene 2, or it's the MDR3 in human, or the ABCB4 mutation, all of which codes for this flippase that is lost and impairs the movement of phospholipids across the canalicular surface, leading to these naked bile salts, and where the cholangiocytes are very, very sensitive to, leading to pericholangitis and periductal fibrosis. The example is shown here about the leakiness and the inflammation within the bile ducts in the MDR2 knockout mice, where these are GFP tagged bile salts, and you can see it leaks in the parenchyma of this particular knockout. But in the genetically competent mouse, you can see that the bile acids stay within the bile ducts. Caveat to the MDR2 includes the lack of development of colitis or bile duct cancer, cholangiocarcinoma. So here are my mice. I'll first show you the conventionally raised MDR2, about 7 to 8 week old. Looks pretty healthy for those that don't look at mice very often. This is a healthy looking mouse. An age-matched germ-free MDR2 knockout mouse looks terrible. It doesn't react very well. It has unkept fur. And if you open up the hood, it has intestinal jaundice. Surprisingly, unlike the Mayo study, in which the MDR2 knockout did not develop any significant clinical disease, on a more aggressive Black 6 background, which I had, there was 100% mortality by 8 weeks. This supports the overall protective role of the microbiota in this model when compared to the SPF of the conventionally raised MDR2, which has an intact microbial, resident intestinal microbial system. This made breeding very, very hard, okay? Furthermore, we went on to validate these observations with early syngenic fecal microbiota transplant or transfer of the germ-free mice, which led to 3/4 of the germ-free mice being protected from the mortality phenotype shown here in orange. But timing is important. This is a time-sensitive mechanism. If we transfer the microbiota, again, from donor MDR2 knockout mice, later than 5.5 weeks, it does not survive. So early transfer is important and may be potentially important when we talk about potential therapeutics. Early on in disease is probably better for overall outcomes. As early as 2 weeks after FMT or fecal microbiota transfer, we are able to see an attenuation of serum total bilirubin, which is a marker of bile duct injury or cholestasis, again suggesting its protective role is towards the liver. Mechanisms, my good man, mechanisms. That's what my PhD PI always told me. So let's get deeper. So what happens when we make the mice germ-free? So in both germ-free MDR2 and wild type knockouts, when we're looking at total bilirubin, total bile acids in the liver, there's a compensatory increase in hepatic bile acid pool. But unlike the wild type, when we look at the serum, unlike the wild type, which is able to appropriately transport bile from the liver, we see here, they both are increased hepatic pools, but transporting the bile acids, the wild type is able to do in a germ-free state. but the MDR2 knockout marked in gray is unable to do so. And this is this is not only a microbial different phenomenon, but also a genetic phenomenon, as we see increases in hepatic bile in the serum bile acid pools in the SPF animals as well. Notably, we also see an elevation in association with worsening cholestasis, shown here with increase in total bilirubin only in the late stage germ-free MDR2 knockout, whereas very early on in the 4 to 5 week period, the germ-free animals not only have decreased bile acid accumulation, but also decreased total bilirubin. And there's no haploinsufficiency, as in the MDR2 heterozygous mice up to 70 weeks do not develop significant hyperbilirubinemia. So we wanted to test this mechanism and validate this mechanism by blocking accumulation of hepatic bile acids. So in normal homeostasis, the terminal ileum takes up to 90 to 95% of fecal bile acids. So to validate our hepatic bile toxic mechanism, we utilize this apical sodium dependent bile acid transporter inhibitor to do so. And so this was done at libidum, where it dissolved in water, and we treated the mice with antibiotics again to develop that compensatory effect versus untreated controls. And shown here in pink are the ASBT inhibitor treated mice, and in green are our controls. And looking at fecal total bile acids, we're able to see a proof of concept where we see increased fecal bile acids in the ASBT inhibitor treated animals, and normal levels in the non-ASB treated mice. And what we also see when we block reuptake, we see a significant attenuation of total bilirubin as well as serum total bile acids, again validating this particular mechanisms as a major driver of hepatic toxicity. So I've been able to show you that the overall role of the microbiota in the MDR2 or in this PSC-murine model is protective. And it not only leads to decrease in inflammation, but what I didn't show you, it also decreases fibrosis, ductular reaction, as well as macrophage recruitment. So the next few slides, I'm going to take you through our journey in figuring out what are the players, the good guys and the bad guys, that leads to these downstream effects in the liver. And so we utilize untargeted metabolomics to identify functional pathways, and the greatest and the most significant pathway were non-surprisingly bile acids. These other pathways, such as zymosterol biosynthesis, nicotine degradation, as well as sucrose degradation, were also seen in wild types, and wild types treated with antibiotics. But the bile acids were, by and large, the biggest increase in effect. And when we treated our mice with selective antibiotics, the antibiotics being vancomycin, neomycin, metronidazole, to target gram-positives, gram-negatives, and anaerobes, we were able to see differential diversity, at least looking at the beta diversity plot here. We're able to see in purple are the vancomycin treated group, which was the most different from our control group in brown. Furthermore, when we looked at in vivo effects of selective antibiotics, it was the vancomycin sensitive population, or the resident microbes, that led to worse outcomes when looking at histologic inflammation, here marked in V, or ductal reaction marked by CK19 staining, quantitated here, or fibrosis gene induction genes such as collagen alpha 1 as well as TIMP1. We saw a significant increase of worsening in disease in the vancomycin-treated group, suggested that this population is where the protective groups are, protective bacteria is mainline. And so of the vancomycin sensitive population that has a lot of literature that shows functionalities that play a role in bile acid metabolism, the family of Clostridiaceae as well as Lactospiraceae are high amongst them. We then took a consortium of from Kenya Honda 16 Clostridial species that we treated our mice with, and we had no response. I failed to also talk about this particular chart where it looks at antibiotic sensitivity. Both Clostridiaceae as well as Lactospiraceae are exquisitely sensitive to vancomycin, whereas Clostridiaceae seem to have a more pleiotropic effect from the other antibiotics, but Lactospiraceae seem to have the most significant effect. So we then utilized a consortium of Clostridia. We had no in vivo effects, but in Lactospiraceae, we were able to see some putative protective effects, which I'll show you in the next few slides. What we did here, based on the experimental setup, we treated the MDR2 from the SPF colony with antibiotics. We had a washout period, and then we added 21 strain of Lactospiraceae versus control, and we looked at various outcomes. We were able to see a decrease in liver inflammation, ductal reaction, as well as liver histologic fibrosis. Furthermore, we wanted to look at other things the Lactospiraceae produce, and one of the major fermenters of short-chain fatty acids are Lactospiraceae, the other being Ruminococcus. And what we saw here when we utilized selective antibiotics, again, V representing vancomycin, N for neomycin, M for metronidazole versus water control, is that vancomycin had the most significant effects in the short-chain fatty acids, which are represented by the major pools of acetate, propionate, as well as butyrate. We then took that to test the effect of short-chain fatty acids alone and independent of Lactospiraceae by co-treating the SPF and mice, MDR2 mice, with vancomycin to, again, eliminate the bacteria, but also then adding short-chain fatty acids. And what we're able to see is significant improvement in bridging fibrosis in the short-chain fatty acid treated MDR2 animals, and overall decrease in liver fibrosis, as well as fibrosis gene induction genes, collagen alpha 1, as well as TIMP1. To further validate this and to make sure that it's coming from the Lactospiraceae, we utilize mass spec to separate of the 21 various species of Lactospiraceae, the high producing versus low producing short-chain fatty acid producing Lactospiraceae. We then treat, and of which there were 3 in each group. We treated, after antibiotic pretreatment, we added the high and low group, and we were able to see again that it was the high short-chain fatty acid producing Lactospiraceae that led to decrease in liver fibrosis, but with no change in liver inflammation. With future directions, looking at downstream targets of these short-chain fatty acids, butyrate and propionate are also HDAC or histone deacetylase modifiers. And so that could be a potential downstream target, or more directly binding to macrophages that express their receptors GP41 as well as GP43 that can lead to effect on stellate cell activation. And we can do that by either modifying genetically receptor knockouts with the MDR2, or by mutating the actual Lactospiraceae. and that's in the future. But what we are able to at least find from our studies is that lactose production of short chain fatty acids seem to have anti-fibrogenic properties in this murine model. But Lacnoman is not done yet. Lacnoman has other mechanisms that we came to found out. And so represented here is a differential abundance of our fecal 16S analysis over time, in which we start with the major populations here in the untreated day 0 group here. We then treated both groups with antibiotics pre-treatment, and what we see is an enrichment of not only pathobionts, Enterobacteriaceae, as well as Enterococcus, seen here in gray and green, but also Ruminococcus. After we, during the washout period and water alone control, what we see is an immediate decrease in Ruminococcus down to baseline levels, suggesting that Ruminococcus is a bystander. But what we see here is a significant expansion of this green group, or this Enterococcus group, that we went on to sequence and find out that it's E. faecalis. But what we see in the lactose-treated group in the last 7 days is that we were able to see an attenuation of this expansion, suggesting an inhibitory role of lactose-Sclerosing on E. faecalis, of which we were able to verify by qPCR that lactose-treated mice, when looking at the fecal microbiota, we saw an attenuation of Enterococcus. We went on to look at this further by looking at whether or not liver translocation was seen, and so I cultured, following the pre-treatment, as well as either treating them with lactose-Sclerosing or media control, culturing the liver. We saw increased translocants in the liver of the control group, but not in the lactose-Sclerosing group. And when we sequenced them, 60% of them were E. faecalis, the other 40% were E. coli. And we didn't see that in the lactose-Sclerosing group. This was mediated by increased intestinal permeability in the MDR2 treated with antibiotics, at least when compared to the wild type control, when utilizing this oral fitz-dextran permeability assay. Furthermore, we took the liver translocants from the MDR2 knockout mice, we then subcultured them, and then gave 10 to the 8th to germ-free MDR2 knockout mice to assess whether or not the translocation was reproducible. And we had 100% of the mice had liver translocants, both E. coli and E. faecalis, and as well as worsening liver inflammation. Furthermore, this translocation, we believe that the translocation and inflammation led to increased mortality, shown here in purple, compared to our germ-free knockout mice, which die at 8 weeks. We saw death as early as 5 weeks in our dual colonized MDR2 mice. And then when we added lactose-Sclerosing to our dual colonized, we found survival protection, suggesting again that lactose-Sclerosing may mediate its protective effects by blocking E. faecalis and Enterococcus translocation. Furthermore, E. coli and E. faecalis also worsens and accelerates hepatic bile acid accumulation and cholestasis, shown here. We're at 4 to 5 week old animals at which nearing death, we were able to see high amounts of serum total bile acids compared to our age-matched controls, the level of serum total bile acids is similar to our 8-week-old germ-free animals that, again, that are very, very sick. And that's also supported by level of cholestasis seen by total bilirubin. So Lacnoman not only has anti-fibrogenic roles, but also has roles in protecting against pathobionts that contribute to accelerated liver mortality. Lastly, analysis of metagenomic data from a multinational Scandinavian repository that we partnered with PSC fecal samples identified enrichment of E. faecalis as well as Enterobacteriaceae and depletion of Lacnospiraceae in PSC patients compared to healthy controls. Furthermore, these pathobionts positively and Lacnospiraceae negatively associated with PSC clinical severity marked by Mayo risk score. And then we also found that E. faecalis were more commonly detected in patients, in PSC patients and controls, here around 17% compared to 5% in healthy controls, which was amplified by in patients who received antibiotics in the last 6 months. This may explain sort of the large heterogeneity in clinical studies involving PSC patients and antibiotics. So in summary, we found an overall protective role of resident microbiota in our MDR2 knockout model, and that antibiotic-induced dysbiosis of SPS MDR2 knockout mice potentiated hepatobiliary disease with hepatic translocation of E. faecalis and E. coli and driving up hepatic bile acid accumulation. And then microbial manipulations and metabolomic analysis in MDR2 knockout mice identify hepatoprotective Lacnospiraceae that inhibited pathobionts E. faecalis, E. coli, hepatic translocation, and mediated anti-fibrotic effects by producing short-chain fatty acids. And in our human cohorts that I just showed you, fecal E. faecalis Enterobacteriaceae positively, and then Lacnospiraceae negatively associated with PSC clinical severity measured by Mayo risk score. And this is a cartoon that I won't go into greater detail about unless there's questions, showing summarizing what we saw. So what do I want to do with this sort of information in my preclinical model? Well, I'm hoping that this gives us a launching point to test human stool using my germ-free knockout mice as a model. And what several studies have shown is that that resident microbiota is relative, that just taking donor stool from random folks and assuming that that's the same population that can coexist in a diseased host may be an error. And so what I plan is, by looking at asymptomatic PSC, which has been estimated to be 10 to 15% of patients that are asymptomatic, defined based on this criteria of both clinical and biochemical outcomes without evidence of portal hypertension, mild stricturing disease, and no exposure to antibiotics, compared to more mild to moderate PSC, taking their stool and reassessing and assessing whether or not my germ-free MDR2 mice can recapitulate disease. And by assessing, prior to that, assessing both metagenomic and metabolomic analysis, and then using that data to assess and stratify candidate microbes and metabolites that we can utilize for future study, with the goal of creating Mu's PSC microbial-based precision medicine, where we can then be able to then take our PSC, mild to moderately diseased PSC patients, assess both the feces, bile, based on metagenomics, if they have an abundance of pathobionts, maybe utilizing selective antibiotics. If they have a low abundance of protective bacteria like Lacnospiraceae, they may benefit from specified donor FMTs or selective consortiums or with and without metabolites to hopefully have some role of influencing therapeutics or finding some utilization for biomarkers. So these microbial insights, I hope, have the potential to better predict clinical disease courses in individual patients, improve PSC fecal transplant outcomes by matching donor and recipient selection, and guide selective microbial manipulations for personalized therapeutic approaches. So in the last sort of few minutes, I wanted to just take you through my PSC-IBD model. But first, I wanted to tell you about the phenotypic differences between PSC-IBD and that of our classic IBD. Notable differences are much more right side predominant disease, decreased activity in the rectum where they have rectal sparing. Both dysplasia and colitis are much more prominent on the right side or the proximal colon. And there's some subset of patients that have increased backwash ileitis, not to be confused with actual ileal disease. And I told you about the increased risk of colorectal dysplasia. And so our models we made the PSC-IBD model by crossing the MDR2, which I talked to you about, with the IL-10 knockout mice, which is a well-known colitis model. It develops spontaneous colitis and is dependent on resident microbiota, shown here, this photomicrograph. And so in my double knockout, the MDR2 IL-10 double knockout marked in red, when we're looking at fecal lipocalin as a marker of colitis, we looked longitudinally over time, and we see early robust fecal lipocalin activity in a double knockout relative to the IL-10 control marked in black, and that plateaus around 10 weeks. And that has been validated histologically, shown here, where at the 6-week time point, we see significant increase in the double knockout relative to the IL-10 control. But what shouldn't be lost is where most of that increase is within the proximal colon. That's compared, and our controls are the MDR2 that don't develop colitis and our wild type controls. By 16 weeks, we're able to see a normalization, at least a relative overall no difference between the IL-10 and the double knockout, but there is a segmental difference regarding the proximal colitis, which is still increased in the double knockout mouse. And that has carcinogenic associations. As early as 16 weeks, we're able to see dysplastic lesions in the proximal colon of the double knockout, and frank mucinous adenocarcinoma by 44 weeks in our double knockouts that we didn't see in our IL-10 knockout. And that's a very strong association here with dysplasia and inflammation. So in conclusion, double knockouts have accelerated pancolitis and early dysplasia relative to IL-10 black 6 mice. Similar to PSC-IBD patients, the predominant proximal colorectal dysplasia in double knockout mice versus IL-10 suggests a conserved mechanism involving concomitant chronic cholestatic liver disease. So our future work will focus on fetal metagenomics and metabolomic analysis to identify protective and inflammatory and potentially carcinogenic species in microbial products driving this disease in this model. So with that, I wanted to thank a lot of people, including both my research and clinical mentors. And I can't tell you how much I have to thank this guy, Balfour. I came in with these wild ideas and he said, prove it, okay? And just giving me the flexibility to do things has been tremendous, and his mentorship is truly a treasure. And again, you can't get far without help. And with that, I certainly want to thank Billy No, our superstar future grad student, as well as Amba Viswanathan. She's going to medical school. And so with that, I'll take questions. These are two of my kids. One that likes pipetting, the other is hiding from pipetting.
All right. Well, Mu, that was a wonderful presentation showing 5 years of research, and so I really want to commend you for your again your innovation and sticking with model, and these are things that I think will have long-term benefits to the field. So I know this area way too well, so I want to open it to other questions. Please, you can either raise your hand. I can't see everybody at a given time, so maybe you should just blurt out stuff.
Alfred, I have a question. Mu, that was great. Great work. It's never easy to start something new. Although I think I may compete with you as the longest serving fellow here. Anyway, so the lacno work is really interesting and how it inhibits E. faecalis. But there's a common theme that I'm seeing with lacno and how it impacts intestinal disease. I'm seeing it in some of Balfour's IL-10 colitis models. I'm seeing it in some of Jenny's NLRP models. We're seeing it in terms of our association with postoperative recurrence. How is this specific to PSC then? Is this something that we are seeing in the context of general intestinal disease? And how do you plan to dissect that? Your human data had controls in PSC, but you didn't have ulcerative colitis patients that didn't have PSC. My feeling would be that they would potentially have the same decrease in lacno and increase in some of the E. faecalis and E. coli that you see in PSC. So I'm trying to understand how you plan to discern that. Is it just a common thing that we're seeing, or is it specific to PSC? And because that has implications, because do you really need to do all the future work you're describing? Why not just start giving people lacno? Like, what is the harm in giving them more lacno without measuring a compensatory decrease in the other bacteria? So I'll just shut up and let you answer, but thank you. Great work.
No, that's a wonderful question. So lacnospiraceae certainly, as you mentioned, may have pleiotropic effects. And to answer your question specifically about a PSC, at least regarding liver disease, lacnospiraceae lack of abundance, or at least decrease, is I've only seen most prominently in PSC. To compare with NASH, to compare with alcohol, which are other prominent diseases where lacno seems to be more enriched, or at least not a major difference. And the reason why I say that with such strength is that the largest metagenomic study was done in that gastro study, and that was highlighted there, versus in several of the metagenomic analysis in alcohol and NASH, there hasn't been a significant decrease in the other etiologies. And so not saying that it doesn't have a role in intestinal disease. I'll actually argue that may be the link, maybe a link of why PSC and IBD are so intricately nested, and that we have to dig in deeper and figure out what these commensal organisms are. But to get to your point, the reason why we can't just willy-nilly give it is that people have tried it. They've tried to give bifido and lactobacillus to PSC patients. And because actually bifido and lacto aren't resident microbiota in PSC patients, it doesn't work. It doesn't work diddly squat. And so I think, at least with PSC field, we have tons of associations, but lack of functionality. And that's where we need to get to the level, the granularity, where we can actually find mechanisms that we can then strengthen and actually have therapeutic effects, rather than just throwing poop on the wall and hoping it sticks. Sorry to use that euphemism, but that's how it seems to be.
Thank you.
All right, other comments? I don't see anything in the chat.
I have a question.
Yeah.
You have a great way with words, Mu. That was a very good simile to use. So I think this ties into what Shizad was just saying. And sorry if I'm beating a dead horse, but like you talked about the lacno, and I guess the short chain fatty acids. Do you think that the utility of this is limited to immunomodulation? Through the, epigenetic modification, or do you think that in the liver also, these short chain fatty acids might be undergoing, leading to some metabolic reprogramming in hepatocytes or other hepatic tissues?
You got my next thoughts, and actually what I'm conducting now, whether or not, so the question is regarding the downstream effect of short chain fatty acids, whether the epigenetic roles or maybe more direct effects on innate immune modifying cells such as macrophages. And I think there's data, at least most of the data is within the intestine. The liver is less known. And the first step is to figure out which of the three, or if there's some synergism regarding a short chain fatty acid. So is it acetate? Is it butyrate? Is it propionate that's driving the anti-fibrogenic role? And that may help us with a mechanism, because acetate doesn't really affect the HDACs versus propionate, and mostly known as the butyrate effects. And then looking at downstream signals, and so we do have primers for the major receptors, and looking at liver expression of that downstream effect to see if there's a signal. That will give us at least a starting point to see what major mechanisms, downstream mechanisms that utilize and have its anti-fibrogenic properties.
Gotcha.
Does that make sense?
Yeah. I mean, I know there was a lot of just like, think about what's next or whatever. But yeah, because if I remember correctly, I think acyl-carnitine pools are the highest in the liver. And that's what's really important for conjugation with the short chain fatty acids for beta oxidation. And that kind of caught my eye a little bit. But yeah. Anyway, thanks. That was an awesome talk.
No worries. Thank you for the question.
Yeah. So Gabriel Suarez has a question. Gabriel, do you want to just say it, or you want me to read it? All right. With a lack of a voice, I'll read it. So he thinks you have incredible epic work here. So congratulations. So and he loved the term lacno man. He had a question regarding, you didn't show data. He's seen your data before. Yeah. So he's asking about the uracolic acid, and that is produced by lacnoman. And but the broader question is, do you feel that the bile acids are mediating some of their effects by differences in their detergent activity at the gut level and potentially in the liver level, within the bile duct as well? And could you comment on how alterations due to decreased phospholipid excretion may affect the detergent effect, and hence the pathogenicity of bile acids? And if you want to specifically talk about the
uracolic acid. Yeah. So there's a lot of questions loaded in that, and I'll break it down into a couple of things. I think it's been, this MDR2 model has been well studied even back to the early 2000s, and the lack of phospholipids makes the protective nature of micelles, which are the nice little detergents that help to dissolve bile acids, pertinent. And so when you don't have phospholipids, it leads to these hydrophobic salts, and they are quite reactive, particularly to the cholangiocytes, and that mediates disease in the bile ducts. But to answer your questions, because I spent almost a year studying bile acid metabolism in this particular field, and it's all over the place. Yes, in the Lactospiraceae, when we looked at differential bile acid metabolism in the feces, we did see in the lacno-treated animals increase in omega-muricolic acid, which seems to have at least may have some protective roles, but it's really the alpha and beta muricolic acids that block against this orphan receptor called FXR. Long story short, the major signal that I got from my differential bile acid analysis is largely bile salt hydrolase activity that's seen in the non-lacno treated animals, which would suggest maybe some detrimental effects of that. But I think
It's really cool. It's evidenced right in PSC. That's one of the features, increased bile salt hydrolase.
That's correct. And so I saw that, but again, at least regarding mechanisms, it's increase in overall hepatic pool and not specifically one particular bile acids, because at least in the liver, by and large, the majority are primary bile acids, which are cholic acid, and the alpha and beta muricolic acids, which are not seen in humans. And so I think there's more to, just to answer your questions shortly, there's more to do regarding and to know about bile acids and its downstream receptors, either if it's TGR5, FXR, or other orphan receptors that have been discovered. But those are great questions.
That's really awesome. Do you think which came first, the egg or the chicken? In the sense, right? So do you create the environment through like liver malfunction, of course, and bile dysregulation, or and then you invite the pathogens, right? Or do you get the pathogens induce the environment? In this case, it seems, my other question was really, are there any host associated mutations in the case of PSC, right? No,
they are. It's polygenetic. A lot of it is seen in a lot of autoimmune diseases, and so it's hard to make a PSC specific model. And so that's the reason, and it explains less than 10%. So the environment, particularly the microbiota, may be the key that may trigger that, similarly to what we see in IBD.
Really cool.
We'll have one quick question, an idea. I don't know if you've done this or not, but have you taken the MDR2 BMMs and just put LPS in there and see what they do? Like, are they more pro-inflammatory? Has everybody injected LPS into these mice? Do they die quicker? It might really help this theory of yours.
Yes, they do. I did one better. I injected it into the portal vein, and he died almost immediately.
Oh, great!
But it destroyed my mice, so I stopped doing it. So, yeah, but to your point.
Yeah, but did the wild type die?
Not as quickly. Not as quickly.
Yeah, but I think the BMM experiment, if you were to do it ex vivo, and if you suddenly find more pro-inflammatory secretions in the absence of MDR2, you're set.
Yes.
You can propose all these butyrate experiments for your K. It's like a perfect sub-aim.
Yes.
Yeah. And I would just add in, Mu, that regarding the general concept of taking either Cooper cells or other cells from the liver and stimulating them with short chain fatty acids, I think that's a huge opportunity. And I agree with Shazad. Do you think because your short chain fatty acids predominantly affected fibrosis, that stellate cells might be another target for short chain fatty acid? And has anyone looked at short chain fatty acid protection in stellate cell function or fibrogenesis?
No. But I know that they don't express, they're not a major expressor of the short chain fatty acid receptors, but it doesn't mean that they don't have epigenetic roles that drive protection. And so that's something I certainly want to explore.
All right, Gabriel, one last question, then we're gonna hang it up.
I just love the subject. So, and I love this guy. So to me, the fact that MDR2 mouse knockouts don't develop colitis spontaneously, right? And you know, to me that kind of suggests that even though you have this bile acid dysregulation, as you see it in IBD also, right? Both in Crohn's and in colitis, is there anything that we know about the abundance of or this dysregulation? It seems to me that it suggests that there's some sort of like localization, right, to the dysregulations, right? So so in the case where you have a coming from, you know, PSC from liver disease, maybe it just it doesn't make it all the way to the stool equally as we see in IBD, or is it something that's different as we see in IBD? Where are you measuring the bile acids?
Yeah, so great question. I think we have to get to a place where we understand the enterohepatic circulation. So what I've done is measuring bile in the stool, liver, as well as in the serum. Those are the major compartments. When you look at bile acid, differential bile acid analysis, comparing the gallbladder to the liver, they're very, very similar. So you can pick one, and the feces is distinct, serum is distinct, and then urine is the only other major compartment. So I chose 3 to give me a sort of assessment of what steady state level looks like. And that's what that's how I was able to do the ASBT experiments and at least support that hypothesis.
Yeah, but Mu, in some of your work, did you compare stool and cecal contents, given the fact that at least in your double knockout, you have a right-sided colitis and dysplasia phenotype?
Yeah, I have, in my initial work, I did cecal contents because that was the most available. But in most of my more recent work has been of the intestinal bile acid pool. I've looked at feces, but looking at a lot of at least studies, there's very little difference between fecal and cecal bile acids, but there hasn't been a lot of work done on it. So maybe there are, but it's certainly in the double knockout, I'll be looking primarily in the cecal and looking at feces and looking at those differences.
Yeah, and the parting advice from Salary Kashiwagi is don't forget you've already mentioned this.
Yeah, GPR 109, that's right.
Receptors in Kupffer cells. So
thank you, Salary.
You know, I think tremendous presentation, Mu, and I think I think your dilemma is not that you don't have enough data for a K award. You got to decide which right. model, which path you
right.
want to take, and any of these could be successful. So I really commend your work, your innovation, and your hard work going into many nights where you and I had very fruitful discussions, but kept each of us from going home to dinner with our families. So anyway, thanks so much.
Thanks, guys. Thank you so much for the wonderful questions. Thanks for the audience. Thanks, guys.